Hello, this is Zach Bird, and this is my presentation of Lab Report 2. In this report, I will discuss a fermentation experiment. I will analyze the data recorded in the experiment, explain the chemical formulas responsible for both cellular respiration and fermentation, and I will answer general questions about the subject matter. My goal is to prove my knowledge of the subject matter and effectively offer guidance to any layman interested in cellular respiration. What is cellular respiration? Cellular respiration is the process in which cells convert glucose and oxygen into ATP. Cellular respiration happens in three stages: glycolysis, the Krebs cycle, and the electron support chain. Reactants: glucose and oxygen. Products: carbon dioxide, water, and ATP. What is the chemical equation for cellular respiration? This is it listed here. Is a process aerobic or anaerobic? Cellular respiration is primarily an anaerobic process. This is because the process requires oxygen to effectively produce ATP. What is fermentation? Fermentation is the process in which cells break down glucose into ATP in the absence of oxygen, also known as anaerobic respiration. Reactants: glucose, products, lactic acid, and ATP, or ethanol, carbon dioxide, and ATP. What is the chemical equation for fermentation? This is it listed below. Is this process aerobic or anaerobic? This process is anaerobic because it does not require oxygen to produce ATP. Instead, it uses the ETC. How does cellular respiration differ from fermentation? Cellular respiration is more efficient and requires oxygen to break down glucose into ATP. Fermentation is a less efficient process that does not require oxygen to break down glucose into ATP. It also produces byproducts. What types of organisms carry out fermentation? Bacteria, fungi, animals, and protists. What was the purpose of this experiment? Develop my knowledge and understanding of cellular respiration and fermentation, while also developing my understanding of what products produce which. How do glucose, sucrose, and starch differ? Glucose is an immediate energy source for cells that is singular sugar unit. Sucrose is made of glucose and fructose and must be broken down before being used for energy. Starch is a polysaccharide composed of glucose. It serves as a long-term energy storage molecule. Why do we use water in our experiment? Water is useful as a stable control variable. It dissolves reactants, supports fermentation, and is stable at room temperature. What was your hypothesis? If the solution has a smaller molecule chain, then it will break down before those with a longer molecule chain. Procedures. Step 1. Label 3 large test tubes, glucose, sucrose, and starch. Step 2. Add 4 milliliters of yeast solution to 3 small test tubes. Step 3. Fill the remaining space in the small test tube with the solutions: glucose, sucrose, or starch. Step 4. Cover the small test tubes with a correlating large test tube. Step 5. Flip tubes and measure the height of bubbles within the small tube. Step 6. Place the tube rack in incubator. Step 7. Record the amount of gas produced in the table. Results. This is a brief table explaining the rate in which the reactants occurred. The glucose reacted first and the starch reacted last. This is because glucose is a simple monosaccharide and the starch is a complex polysaccharide. Which carbohydrate solution fermented the fastest? The glucose. Glucose is a monosaccharide and thus reacted the fastest. Glucose is a monosaccharide, so it is easier to break down. The starch fermented the slowest. The solution did not begin fermentation. It is a polysaccharide and much more difficult to break down. Yes, the sugars with a shorter molecular chain broke down faster than those with a longer molecular chain. The results supported my hypothesis. The sugars with a shorter molecular chain broke down faster than those with a longer molecular chain. Changes could be made to the experiment to add cell respiration. This could contribute to a larger data table and create a more thorough experiment. These are the primary sources used in my lab report. Thank you.